In this question, you have to identify a structure in an angiosperm that develops from the megaspore. Option A, endosperm. Now, the endosperm develops after fertilization when one of the male gametes fuses with the central cell of the embryo sac, but it does not arise from the functional megaspore. Hence, it is incorrect. Option B, embryo sac. The functional megaspore undergoes mitotic divisions to form the female gametophyte, also known as embryo sac. Hence, it is the correct option. Option C, embryo sac. This is incorrect because the embryo develops after fertilization when the zygote undergoes division, but it is not formed directly from the functional megaspore. Option D, ovule. It is also incorrect because ovule is a structure of the plant that contains the megasporangium, which produces the functional megaspore. But the ovule itself does not develop from the functional megaspore. In this question, you have to identify now which type of pollination or flowers requires attractants and rewards. Option A, entomophily. Now, entomophily refers to pollination by insects, and attractants like bright colored flowers, different patterns, and scents are very important for attracting insects. And rewards such as nectar and pollen serve as an incentive for the insects to visit the flowers again and again, thus facilitating pollination. Hence, this is the correct option. Option B, hydrophily. Now, this is incorrect because hydrophily involves pollination through water. So, they do not require any attractants like bright color or scent, nor they require rewards like nectar. Option C, cleistogamy. This is also incorrect because it refers to self-pollination that occurs in closed flowers. Option D, anemophily. It is also incorrect because anemophily refers to pollination by wind. And wind pollinated plants, they produce large quantities of lightweight pollen, but they do not attract pollinators or provide rewards. In this question, you have to identify what type of fruit is coconut. Option A, berry. It is incorrect because a berry has a fleshy pericarp with the seeds embedded within, like grapes, then tomatoes. But coconut is not such type of fruit. Option B, nut. It is also incorrect because a nut is a one-seeded fruit which cannot be split open. Option C, capsule. It is also incorrect because a capsule is a dry fruit that splits open to release seeds, such as in cotton or in poppy. But coconut doesn't have such characteristic. Option D, drupe. Now, coconut is a drupe fruit because it has three distinct layers in its pericarp: an outer exocarp, a fibrous mesocarp, and a stony endocarp that encloses the seed. So this is the characteristic of drupe fruits such as coconut as well as mangoes, peaches. Hence, it is the correct option. In this question, you have to identify the tissue which arises from the vascular cambium. Option A, primary phloem. It is incorrect because primary phloem is formed by the apical meristem during the primary growth and not by the vascular cambium. Option B, secondary xylem. The vascular cambium is the lateral meristem, which is responsible for the secondary growth, and it produces secondary xylem, that is wood, on the inner side and secondary phloem on the outer side. And this activity increases the girth of the plant stem or root, and it contributes to the thickening of the plant over time. And this process is also very prominent in woody plants, and it leads to the formation of the annual rings in trees. Hence, it is the correct option. Option C, periderm. It is incorrect because periderm is a part of the protective tissue that is formed during secondary growth, but it is produced by the cork cambium, that is phelogen, and not the vascular cambium. Option D, pheloderm. It is also incorrect because pheloderm is a tissue that is produced by the phelogen and not the vascular cambium. In this question, you have to identify what facilitates The opening of stomatal aperture. Option A, decrease in turgidity of guard cells. Now, a decrease in turgidity of guard cells causes the guard cells to collapse, leading to the closure of the stomata and not its opening. Hence, it is incorrect. Option B, radial orientation of cellulose microfibrils in the cell wall of guard cells. Now, the radial orientation of cellulose microfibrils in the guard cell walls allow the cells to expand in a way that the stomatal aperture opens. And this radial orientation of cellulose microfibrils is indeed very important because this arrangement ensures that when the guard cells become turgid, they elongate rather than expanding uniformly in all directions. And this elongation causes the stomatal pore to open. Hence, it is the correct option. Option C, longitudinal orientation of cellulose microfibrils in the cell wall of guard cells. It is incorrect because if the cellulose microfibrils were oriented longitudinally, the expansion of the guard cells would not result in the opening of the stomatal pore because this arrangement would restrict the outward expansion. Option D, contraction of outer wall of guard cells. It is also incorrect because the outer wall of the guard cells does not contract. Instead, it stretches and bulges outward when the guard cells become turgid, thus helping in the stomatal opening. In this question, you have to identify that in Bougainvillea, thorns are modifications of which part? Now, in Bougainvillea, the thorns are modifications of the stems that develop from the axillary buds. And these structural modifications may help to protect the plant from herbivores. Hence, the correct answer is option B, that is. In this question, you have to identify that root hair develops from which region of plant. Option A, elongation. It is incorrect because the region of elongation in the root is where the cells increase in size, but root hairs do not develop here. Now, this zone is rather responsible for the lengthening of the root. Option B, root cap. It is also incorrect because the root cap is the region that protects the root tip as it grows through the soil, but it does not give rise to root hairs. Option C, meristematic activity. It is also incorrect because the region of meristematic activity is where the cells actively divide, but root hairs do not form from here. Option D, maturation. Now, root hairs develop from the region of maturation, that is, where cells differentiate and specialize. And this zone is involved in the absorption of water and nutrients from the soil. And that's why root hair play an important role. Hence, it is the correct option. In this question, you have to identify which of the following is made up of dead cells. Option A, collenchyma. Now, it is incorrect because it is made up of living cells with thickened cell walls. Option B, phloem. Phloem, which is also known as cork, is made up of dead cells. And these cells are filled with substance called suberin, which is a waxy material that makes the cell walls impermeable to water and gases. So it forms a protective layer of the plant, particularly in woody plants. And it protects the plant from desiccation or pathogens and even mechanical injuries. Hence, it is the correct option. Option C, phloem. Now, it is incorrect because it is composed mostly of living cells, such as the sieve tube elements and the companion cells. But some components, like the phloem fibers, are dead. Option D, xylem parenchyma. It is also incorrect because xylem parenchyma consists of living cells. In this question, you have to identify the colonial alga. Option A, volvox. Now, volvox is a colonial alga where individual cells form a colony and they work together, exhibiting a division of labor. Hence, it is the correct option. Option B, eulothrix. It is incorrect because it is a filamentous alga and not colonial. Option C, Spirogyra. It is also incorrect because it is also a filamentous alga and it is even characterized by spiral chloroplasts. Option D, Chlorella. It is incorrect because Chlorella is a unicellular alga and it does not form colonies. In this question, you have to identify the plants which produce pneumatophores and show vivipary. Option A, halophytes. Now, halophytes are salt-tolerant plants found in coastal and saline environments. They produce pneumatophores, that is aerial roots for gaseous exchange in waterlogged conditions. And they also exhibit vivipary, that is seeds germinate while still attached to the parent plant. And both these adaptations help them to thrive in harsh environments. For example, mangroves. Hence, it is the correct option. Option B, samophytes. It is incorrect because samophytes are plants adapted to sandy soils, such as those in deserts. But pneumatophores are absent and they also lack vivipary. Option C, hydrophytes. It is incorrect because hydrophytes are plants that are adapted to aquatic environments. Now, they have adaptations like erenchyma for buoyancy, but they do not have pneumatophores or exhibit vivipary. Option D, mesophytes. It is also incorrect because these are the plants adapted to moderate and non-extreme environments. So, they lack special adaptations like pneumatophores and vivipary. In this question, you have to identify the statement which is not correct regarding the factors affecting the rate of photosynthesis. Option A, increasing atmospheric carbon dioxide concentration up to 0.05% can enhance carbon dioxide fixation rate. Now, this is true because increasing atmospheric carbon dioxide levels up to 500 ppm, that is 0.05%, can enhance the rate of photosynthesis, particularly in C3 plants. Now, this is because the enzyme Rubisco in C3 plants operates most efficiently at higher carbon dioxide concentrations, thus reducing photorespiration and increasing carbon fixation. Hence, it is incorrect option. Option B, C3 plants respond to higher temperatures with enhanced photosynthesis, while C4 plants have much lower temperature optimum. Now, this is not correct. C3 plants have a lower temperature optimum compared to C4 plants because C4 plants are more adapted to higher temperatures and they show increased or enhanced photosynthesis under such conditions due to their specialized carbon fixation pathway, which minimizes photorespiration. But in contrast, C3 plants suffer from increased photorespiration at higher temperatures, thus reducing their photosynthetic efficiency. Hence, it is the correct option. Option C, tomato is a greenhouse crop which can be grown in carbon dioxide enriched atmosphere for higher yield. Now, this is also true. Tomatoes, which are C3 plants, they can benefit from CO2 enrichment in the greenhouse conditions. So, higher carbon dioxide conditions can lead to the increase in the rate of photosynthesis, thereby boosting their growth as well as the yield. Hence, it is incorrect option. Option D, light saturation for carbon dioxide fixation occurs at 10% of full sunlight. This is also true because light saturation for photosynthesis in most plants indeed occurs at 10% of full sunlight. And beyond this level, increasing light intensity does not enhance the rate of photosynthesis significantly because of other factors such as carbon dioxide availability or enzyme activity, which becomes limiting. Hence, it is incorrect option. In this question, you have to identify the phytohormone, that is the plant hormone, the application of which can prevent fruit and leaf drop at early stages. Option A, ethylene. It is incorrect because ethylene promotes fruit and leaf drop rather than preventing it. Option B, auxins. Now, auxins, particularly the synthetic auxins like the NAA, that is naphthalene acetic acid, and 2,4-D, that is 2,4-dichlorophenolic acid, prevent premature fruit and leaf drop by maintaining the strength of the abscission zone and promoting cell elongation and division. Hence, it is the correct option. Option C, gibberellic acid. It is incorrect because gibberellic acid promotes stem elongation, seed germination, and flowering, but it is not effective in preventing fruit and leaf drop. Option D, cytokinins. It is also incorrect because it delays senescence by promoting cell division, but they are not very specifically effective in preventing fruit and leaf drop. In this question, you have to identify that zygotic meiosis is characteristic of life cycle of which organism? Option A, fucus. It is incorrect because in the life cycle of fucus, gametic meiosis is observed during gamete formation, but not zygotic meiosis. Option B, funaria. It is also incorrect because in their life cycle, meiosis occurs during spore formation in diploid sporophyte, but not in zygote. Option C, Chlamydomonas. Now, certain species of Chlamydomonas exhibit zygotic meiosis. That is, the zygote is the only diploid stage, and meiosis occurs immediately after zygote formation to produce haploid cells, characteristic of organisms with a haplontic life cycle. Hence, it is the correct option. Option D, Merchantia. It is also incorrect because in the life cycle of Merchantia, meiosis occurs during spore formation in the diploid sporophyte, but not in zygote. Flowers which have single ovule in the ovary and are packed into inflorescence are usually pollinated by option A, bee. It is incorrect because flowers pollinated by bees usually have multiple ovules and are not densely packed into inflorescence. Rather, these flowers are bright colors, nectars, and fragrance. Option B, winged. Now, flowers with a single ovule in the ovary and densely packed into inflorescence are usually adapted for wind pollination because these flowers are small, inconspicuous, and they do not have nectar or fragrance. Hence, it is the correct option. Option C, bat. It is also incorrect because bat pollinated flowers are generally large and they produce a profuse amount of nectar with strong fragrances. Option D, water. It is also incorrect because water pollinated flowers are usually aquatic and they have specific adaptations for pollinations through water. Now, which is not related to the single ovule flowers in dense inflorescence. In this question, you have to identify what type of life cycle do ectocarpus and fucus respectively have. Now, ectocarpus, which is a brown algae, exhibits a haplodiplontic life cycle. That is, it has both haploid and diploid multicellular stages. The haploid gametophyte produces gametes and the diploid sporophyte produces spores via meiosis. And both these stages are morphologically similar. And fucus, which is also a brown algae, exhibits a diplontic life cycle where the dominant stage is diploid. So the haploid stage is restricted to the gametes only, which are produced by meiosis. And the diploid zygote grows directly into mature individual without a multicellular haploid stage. Hence, ectocarpus exhibits haplodiplontic life cycle and fucus exhibits diplontic life cycle. Thus, the correct answer is option B. A dioecious flowering plant prevents both option A, autogamy, and geitonogamy. Now, in dioecious plants, male and female flowers are born on separate plants. And this separation ensures that autogamy, that is, self-pollination, cannot occur because a single flower cannot produce both pollen and ovules. And geitonogamy, that is, transfer of pollen between different flowers on the same plant, is also prevented because male and female flowers are on different plants. Hence, it is the correct option. Option B, geitonogamy and xenogamy. Now, while dioecious plants prevent geitonogamy, but they promote xenogamy, that is, cross-pollination between different plants to ensure reproduction. Hence, it is incorrect. Option C, Cleistogamy and xenogamy. It is incorrect because cleistogamy, that is pollination within closed flowers, is unrelated to the dioecious flower, and xenogamy is not prevented in dioecious plants. Option D, autogamy and xenogamy. Now, dioecious plants prevent autogamy, but as already stated, they encourage xenogamy. Hence, it is incorrect. In this question, you have to identify the wrong statement regarding Krebs cycle. Option A, there is one point in the cycle where FAD plus is reduced to FADH2. This is true. This occurs during the conversion of succinic acid to fumaric acid, where FAD plus is reduced to FADH2. Hence, it is incorrect option. Option B, during conversion of succinyl-CoA to succinic acid, one molecule of GTP is synthesized. This is also true. In this step, substrate level phosphorylation occurs, resulting in the synthesis of GTP, which can be converted to ATP. Hence, it is incorrect option. Option C, the cycle starts with condensation of acetyl group, that is acetyl-CoA, with pyruvic acid to yield citric acid. Now, this is wrong because the Krebs cycle starts with the condensation of acetyl-CoA with oxaloacetic acid and not pyruvic acid to form citric acid. Pyruvic acid is converted to acetyl-CoA in a separate process before entering the Krebs cycle. Hence, it is the correct option. Option D, there are 3 points in the cycle where NAD is reduced to NAD plus and H plus. Now, this is also true. This happens during the conversion of isocitric acid to alpha ketogluteric acid and during the conversion of alpha ketogluteric acid to succinyl-CoA, and finally during the conversion of malic acid to oxaloacetic acid. Hence, it is also incorrect option. Now, phosphoenol pyruvate, that is PEP, is the primary carbon dioxide acceptor in option A, C4 plants. In C4 plants, PEP is the primary acceptor of carbon dioxide, and this occurs in the mesophyll cells where PEP combines with carbon dioxide in a reaction which is catalyzed by PEP carboxylase, forming oxaloacetic acid, that is OAA. And this mechanism is efficient and it also minimizes photorespiration. Hence, it is the correct option. Option B, C2 plants. It is incorrect because the term C2 plants refers to plants that undergo photorespiration, where glycolate is the significant intermediate, but PEP is not involved as the primary carbon dioxide acceptor. Option C, C3 and C4 plants. It is also incorrect because in the C3 plants, ribulose 1,5-biphosphate, that is RUBP, is the primary carbon dioxide acceptor and not PEP. But it is true for C4 plants. Option D, C3 plants. It is also incorrect because, as already stated, RUBP is the primary carbon dioxide acceptor in the Kelvin cycle. In this question, you have to identify the wrong statement in context of heartwood. Option A, it is highly durable. This is true. Heartwood is highly durable because it is composed of dead lignified cells, and compounds like tannins and resins are deposited in it, making it resistant to decay. Hence, it is incorrect option. Option B, it conducts water and minerals efficiently. Now, this is not true. Heartwood is non-functional for water and mineral conduction as its cells are dead and it is blocked with deposits of organic substances. Hence, it is the correct option. Option C, it comprises dead elements with highly lignified walls. It is true, as already stated, it is indeed made up of dead cells with lignified walls. Option D, hence it is incorrect option. Option D, organic compounds are deposited in it. It is also true. Organic compounds such as tannins, resins are deposited in heartwood. Hence, it is incorrect option. In this question, you have to identify the mismatch pair. Option A, cycas is to dioecious. Now, this is true because cycas is dioecious. That is, male and female reproductive structures are present on separate plants. Hence, it is incorrect option. Option B, Salvinia is to heterosporous. This is also true because Salvinia is heteroporous. That is, producing two types of spores, microspores and megaspores. Hence, it is incorrect option. Option C, Equisetum is to homosporous. This is also true because Equisetum, that is the horsetails, is homosporous. That is, producing only one type of spore. Hence, it is incorrect option. Option D, Pinus is to dioecious. Now, this is not true because Pinus is monoecious. That is, male and female reproductive structures, here they are represented with cones, are present on the same plant. Hence, it is the correct option. In this question, you have to identify the morphological nature of the edible part of coconut. Option A, cotyledon. It is incorrect because the cotyledon is the embryonic leaf within the seed. And in coconut, it is not the edible part. Option B, endosperm. The edible part of the coconut is the endosperm. In its solid form, it constitutes the white kernel, and in its liquid form, it is the coconut water. Hence, it is the correct option. Option C, pericarp. It is also incorrect because the pericarp is the outer covering of the fruit, which includes the exocarp, that is the outer layer, the mesocarp, the fibrous middle layer, and the endocarp, the hard shell. But these are not edible in coconut. Option D, perisperm. It is also incorrect because perisperm is a nutritive tissue that is derived from the nucellus, and it is not present in coconut. In this question, you have to identify the organism that exhibit double fertilization. Option A, algae. It is incorrect because algae reproduce to various methods, including vegetative, then asexual, sexual reproduction, but they do not exhibit double fertilization. Option B, fungi. It is also incorrect because fungi reproduce via spores through sexual or asexual means, but they do not have double fertilization. Option C, angiosperms. Now, double fertilization is a characteristic feature of angiosperms, where one sperm cell fuses with the egg cell to form a zygote from which embryo arises, and the other sperm cell fuses with the central cell to form the triploid endosperm. Hence, it is the correct option. Option D, gymnosperms. It is also incorrect because gymnosperms do not exhibit double fertilization. They have a single fertilization process where one sperm nucleus fertilizes the egg and no endosperm is formed.